Technologies 101 with Lucy. Today our focus is Agrobacterium tumefaciens. This is a bacterium that affects plants and causes what we call crown gall disease. As you can see in this picture, plants usually have tumors that can be caused by this type of bacterium. What happens is that the bacterium infects a wound on a plant, then it will inject a short stretch of DNA into the wound or transfer DNA into the wound, and this DNA usually will come from what we call a plasmid. In the structure of a bacteria, we usually have the genomic DNA and now extra chromosomal DNA in form of plasmids. So this plasmid in this bacterium is called the Ti plasmid, and the Ti usually now will cause what we call tumor induction. Now a short region within this Ti plasmid called the tDNA or the transferred DNA is the one that will be transferred into the plant cell once this agrobacterium infects the wound of a plant, and it will cause the cell to grow a tumor-like structure. So this tDNA contains the genes that allow the infected cells to make unusual compounds such as nopalin and octapine, and these are the ones that are now responsible for the tumor-like structure called a gall tumor that looks like this. Now this bacterium has been utilized in genetic engineering or recombinant DNA technology because of its ability to have transfer of DNA from one organism into the other. You can see that we are transferring a tDNA from the Agrobacterium plasmid into the plant cells, resulting into the formation of a tumor. So that transfer mechanism can be used in genetic engineering or recombinant DNA technology, where now this Ti plasmid is modified to ensure we do not have the genes that code for the tumor, the two genes that code for the tumor, but now we insert our foreign fragment. Maybe there is a certain gene that we want to transfer to a certain plant that has various advantages. Maybe it is for resistance against certain infections. This foreign gene is going to be incorporated into the Ti plasmid, then inserted back to the host bacterium, which is now the Agrobacterium tumefaciens, and then we culture this bacterium with isolated plant cells or the wounded plant tissues that we want to get our foreign gene, and the new gene is eventually injected into the cells and ends up being integrated into the chromosomes of the plant. And therefore, when the plant is having transcription, translation into proteins of interest, then it is able to also transcribe and translate the foreign gene that has various advantages to that plant.